How do we know what we know in science? The scientific process of discovery works much the same way as the detective works. The detective makes an observation and then tries to formulate alternatives that could explain it. It's something curious. They find ways to test it. They perform those tests and it leads them down a new avenue. Science works in much the same way. We observe something in nature that we find curious, and we then try to come up with hypotheses or testable predictions that allow us to determine why things are the way they are. The scientific method involves systematic observation and measurement, the formulation of hypotheses, these testable predictions, and then the conducting of experiments that can exclude one hypothesis or another. The greatest philosopher of science of the 20th century, Karl Popper, put it this way: science may be described as the art of systematic oversimplification. Popper pointed out that a hypothesis or a theory is scientific if and only if it's falsifiable. That is, if and only if it can be disproved. So for example, the tenets of religion are not hypotheses, they're not scientific theory, because they can't be disproved. There's no experiment you could come up with that could disprove the existence of God, for example, and therefore religion does not fall within the realm of science. John Platt formalized this in another way through his idea of strong inference. As Platt put it, a theory that predicts everything does not predict anything. This is not science but faith, not theory but theology. A theory is not a theory unless it can be disproved. Platt had three components to his strong inference idea. In the first part, the scientist devises alternative hypotheses, that is, alternative testable predictions that could explain something of interest. In the second stage, a critical experiment is conducted that could exclude one possibility or the other. After this procedure is recycled in the third stage with sequential hypotheses that can be tested. By doing this process, the scientist works his or her way down a series of experiments that may lead in very unexpected directions. Our experiments can take us down many pathways of discovery. Each time we hit a node or alternative hypotheses, we conduct an experiment that will send us one way or the other. The original scientific philosophers were thinking along these same lines. For example, Francis Bacon said, "To man it is granted only to proceed at first by negatives and at last to end in affirmatives after exclusion has been exhausted." Or as Platt put it, any conclusion that is not an exclusion is insecure and must be rechecked. The most productive scientists in history have really followed this approach. For example, Röntgen, when he discovered X-rays, discovered 17 major properties of X-rays within 8 weeks of his original discovery. This, by the way, is in the first X-ray image that he took. It's of his wife's hand. And the image apparently scared her because her response was, "I have seen my death." Platt said we should try, like Pasteur, to see whether we can reach strong inferences that encyclopedism could not discern. What he meant by that is knowing the most of anyone in the world about a problem doesn't allow you to solve that problem. What allows you to solve a problem is to tackle that problem with the best tools at your disposal, and not to be stuck on the use of any particular tool, but rather choose the tools that best address the problem at hand. In other words, to take a problem-oriented rather than a method-oriented approach. Pasteur was a master at this, and because he took a problem-oriented approach, and with each problem that he tackled, he found the most appropriate tools to test it, he advanced science like no one had in that century. Pasteur solved the problem of the infectious agent of anthrax and came up with the germ theory of disease that is the basis of modern medicine. Pasteur solved the problem of preserving food through what we now call pasteurization. He also tackled the age-old problem of spontaneous generation, and he disproved spontaneous generation in a series of clever experiments. As Pasteur put it, chance favors only the prepared mind. He was prepared not only in his education, which was excellent, but in his method. His method of following the problems, devising experiments that could exclude one possibility or another, and seeing where that led him. This idea of strong inference in the scientific method, it's much like following the trunk of a tree. We move along the trunk of a tree to a branching point, At that branching point, we devise alternative hypotheses for something. We then have to come up with an experiment that allows us to exclude one possibility or the other. Once we've excluded one of the possibilities, we follow the line of logic that the other possibility demands. And we get to the next branching point. And we devise the next set of experiment which allows us to, again, exclude one of the two alternative hypotheses. Popper also pointed out that proof in science does not exist, but rather science advances by disproofs. What he meant by this is that we never know how the future knowledge, the future discovery of science will improve our understanding of the world. We might think that we totally understand something now, and then later we find out that it's only partially the truth. For example, Newton's laws of physics were considered completely established. Every single test affirmed his laws of physics, until Einstein came along and showed that at extreme velocities approaching the speed of light and under extreme gravitational fields, Newtonian physics breaks down. That is, Newtonian physics is a special case of relativity under normal gravitational and velocity circumstances. Einstein showed that what we had thought as proven laws needed to be modified. And in fact, Einstein's ideas may need to be modified in turn in the future. Popper summarized this whole approach this way. He said, whenever a theory appears to you as the only possible one, take this as a sign that you have neither understood the theory nor the problem which it was intended to solve. So how do I take this approach in my own work? Let me give you an example with an environmental contaminant called perchlorate. Perchlorate is a strong oxidizer. It's the oxidizer that we use in solid rocket propellant, powering space shuttle, rockets, even artillery. But perchlorate is also an environmental contaminant. Perchlorate causes hypothyroid condition. It actually prevents the synthesis of thyroid hormone in the thyroid gland. And this is of paramount importance to human health and animal health because thyroid is involved in so many aspects of development. It's particularly risky when an infant or a fetus in the womb is exposed to perchlorate because of its effects on brain development and other developmental pathways. Why should we be concerned about perchlorate contamination? Well, we should be concerned because there are many sources of contamination, and in fact, all of us are exposed to perchlorate in the environment. In this video, we can see what happens on a bad day. 5, 4, 3, 2, 1. 0. We have ignition and liftoff from Cape Canaveral Air Station of the Air Force Delta II launch vehicle carrying the new GPS-2R satellite. We have had an anomaly. And these large chunks of flame that are falling down to the earth are actually pieces of perchlorate that are contaminating the local environment. Such rocket launch failures are more or less common, and as a consequence, we have contamination of the environment from failed launches in many places around the United States and in other parts of the world. But failed rocket launches aren't the only source of perchlorate contamination. In fact, most perchlorate contamination comes from improper storage at production facilities and facilities where perchlorate is used, such as in aerospace and Department of Defense facilities. A recent study showed that out of thousands of Americans tested for perchlorate, 100% had perchlorate in their urine. All of us are exposed to it through our drinking water, through contaminated milk, and leafy vegetables. We decided to see what perchlorate does to development by studying its effects on a model fish species, the three-spined stickleback fish. This fish is, believe it or not, a great model for human health because its developmental pathways are similar to humans. Fish share about 70% of their genome with humans. The hormone system is the same. So by studying how perchlorate disrupts the development of this fish, we can actually gain insights into how perchlorate disrupts development of humans and how perchlorate may cause problems with human health. When we started testing our hypotheses for how perchlorate affects development, we found some curious things. Here, for example, we can see a reproductive male. He has bright red coloration on his throat, bright blue eyes, and iridescent green back. The fish that's exposed to perchlorate, on the other hand, is transparent. It's failed to lay down its dermal bones. It's failed to produce pigmentation. We can actually see right through it and see its internal organs. The most curious thing we found was we had a fish that looked like a female, swollen with eggs, but it was acting like a male, acting with male reproductive behavior. When we opened that fish up, we found that she actually had sperm and eggs. We took some of her sperm and we crossed them in a petri dish with eggs from a wild-caught female. We took some of her eggs, crossed them in a petri dish with sperm from a wild-caught male. We also crossed some of her sperm with her own eggs. In all three cases, you can see the micropyle, the point where the sperm penetrated the egg, the embryo was produced and began to develop. So here was a fish that was actually a hermaphrodite. It produced both sperm and eggs. The first question we had to ask ourselves, is this a female that's been disrupted in its development to become a hermaphrodite? Or is this a male that's been feminized? We know of many contaminants that, when fish are exposed to them, can feminize males into females or males into hermaphrodites. Many pesticides do this, for example. But people had not found an environmental contaminant that did the opposite. That is, it masculinized female fish into hermaphrodites. We tested it with these fish, and in fact, this and other fish like it were female fish, genetically female fish, that were masculinized into hermaphrodites. So this is a contaminant that was disrupting sexual development of females and making them produce both sperm and eggs. This made us concerned about its effects on humans. We've seen in industrialized countries a huge increase in reproductive disorders, and maybe some of this increase is caused by perchlorate. When we looked at this fish's gonads, we found that they're a mix of testicular tissue, the black pigmented cells, and ovarian tissue, the red pigmented cells. So this fish was truly a hermaphrodite, producing both sperm and eggs. As we've studied perchlorate, we found ourselves following this trail of pebbles in the woods, just like Hansel and Gretel, leading to very unexpected places. When we first started out studying the effects of perchlorate, we had no concept that we might end up finding disruption of sexual development. And even when we looked at the thyroid, we found unexpected things. This is thyroid tissue from a normal stickleback fish, large thyroid follicles, perfectly healthy thyroid tissue. Here we can see thyroid tissue of a perchlorate-exposed fish. This fish has proliferated many small thyroid follicles rather than having a few big ones. And when we zoom in on those thyroid follicles, we can see that they're surrounded by blood vessels. There's a proliferation of blood vessels, what's called angiogenesis. And this really caught our eye because we see the same kind of condition in certain kinds of cancer. So with our perchlorate work, we started up our investigations heading up the trunk, heading up to this first branching node. We had a hypothesis that these effects of perchlorate were caused by disruption of thyroid hormone. And in fact, some of them were. But some of them weren't. And that led us in unexpected directions. As we headed up this tree and headed along these branching nodes, we found conditions similar to what we see in cancer. We found conditions of disruption of sexual development. We found many things that we never expected to see. And in fact, each of our discoveries has led us to new questions and new mysteries. If we look at the pace of scientific discovery since 1950, it's truly remarkable. Think about it. In 1950, we still didn't know the basis of genetics. We didn't know the inheritance was due to DNA. DNA had not yet been discovered. Now, just 70 years later, here we are in a situation where it's actually less expensive to sequence your entire genome than it would be to store that information on a server. That's remarkable. Every year, the pace of scientific discovery accelerates. It's exponential. We know more each year by far than we knew the last year, and that pace is constantly increasing. I would love to have Louis Pasteur come to us today to see how science is done. I think he would be amazed at the pace at which science occurs. But the thing that would be the greatest about that is that he would be very satisfied to see that the approach we take today is exactly the approach that he took. We still operate by formulating these testable predictions, hypotheses, and coming up with the most clever experiments to test them, to exclude one possibility and to chase down another.